will actually start. All right. I think we are live. Good morning. I can see there are about 54 of you so far, so good on you. I'm so glad to have you here this morning. Welcome and thank you for joining us today for Dr. Corey Wisner's webinar, Harnessing the Power of Trillions, the Microbiome in Health. Let me introduce myself. I'm Michelle Tuttle. I'm a certified sports dietitian and a Team USA athlete, and I'll be moderating our session today. Now, before I introduce Dr. Wisner, I want to tell you just a little bit about our sponsors of today's presentation. And those sponsors are the Wheat Foods Council and the Center for Nutrition and Athletics. The Wheat Foods Council, for those of you who don't know us, is a nonprofit organization formed almost 50 years ago to help educate people about the value of grains, complex carbohydrates, and fiber as essential components of a healthful diet. The Center for Nutrition and Athletics is a website developed by the Wheat Foods Council that's tailored to personal trainers, fitness professionals, and their clients. And if you aren't familiar with all the resources available on these two sites for registered dietitians as well as personal trainers, I'll invite you to visit these websites and learn more about the e-magazines, infographics, videos, and the app that's available. So just a bit of housekeeping before I introduce Corey. Near the end of today's webinar, you're going to be able to download the CEU certificate in the files section of this platform. So if you look over on the right-hand side of your screen, you'll see that you've got a number of things, including settings, chat, slides, etc. And on one of those settings is the files area, and you'll see that we have handouts, and that's where the CEU certificate will be. Today's presentation is going to last approximately 50 minutes. We're going to have about 10 minutes for Q&A. So please, as we go through, go ahead and chat your questions into the chat area, and I will pull those questions when we get to the end of today's presentation, and I'll moderate the questions to Dr. Wisner. So the other thing just to remind you of is that today's presentation will be available it will be available now, of course, and then also after today, so you can view it at any time. All right, now enough of that. Let me introduce today's speaker, and then I'll turn things over to her. So the first thing you need to understand about Dr. Corey Wisner is that she's both a tenured professor and has an impressive athletic background. Her lifelong fascination with nutrition became personal when she suffered 2 fibula stress fractures as a Division I varsity student athlete on both the Purdue cross-country and track and field teams. Her determination to get back to running through improved nutrition not only resulted in her being a national qualifier for the mile with a career best of 4:45, but it also led directly to her getting a PhD in nutrition science from Purdue. She's also completed a postdoctoral fellowship in the Division of Nutrition Sciences at Cornell University, studying how maternal diet impacts maternal bone health and fetal development among pregnant adolescents. Dr. Wisner is now on the faculty of Arizona State University in the College of Health Solutions, where her research focuses on dietary components that modulate the gut microbiome for disease prevention and sustained health throughout life. So, this is going to be great. And so without further ado, let me turn things over to you, Dr. Wisner, and I'll see you later.
Thank you, Michelle, for that wonderful introduction. And thanks, everyone, for joining us. For me, this is bright and early, but if you're on the East Coast, it's closer to lunchtime. So feel free to grab some lunch or snacks and enjoy the presentation as we learn how to feed all those trillions of bacteria that live inside of our gut and exercise to fuel ourselves and them as well. So to outline briefly what we'll be covering today, I want to just start by introducing the microbiome and talk about different factors that influence how they operate and control and modulate what they do for us. And then we'll think about that in the context of what we eat. So rather than looking at my plate in terms of human health, we're going to think about it in terms of, well, what are those foods providing to the microbes that live in our gut and then allowing them to do more for us? And then we're going to look at exercise, which is a really fascinating emerging area in the field of gut microbiome research. And then I'll tie things up with some practical applications that you can use when working with clients, and then some other takeaway messages for the future. So to get things started, I think having some context about what's important in terms of health literature and clinical practice and working with clients in a variety of different health sectors is that most of us are working towards either preventing or treating cardiometabolic diseases. And so it's probably no surprise that obesity is a major public health problem, not only in the United States, but also globally. And once someone becomes obese, they are more likely to suffer from a number of other cardiometabolic diseases like cardiovascular disease, type 2 diabetes, cancer, and metabolic syndrome. And this is quite costly within the healthcare system, as well as on individuals, especially if they have minimal access to healthcare resources and funds for insurance. And so this is a pervasive problem that's been going on for a long time, and we know that lifestyle factors have an important role in mitigating the risk for cardiometabolic diseases. So we can intervene with diet, exercise, but we also now know that the environment in which you live in also impacts those outcomes. And for a long time, we've known that lifestyle can change metabolic signaling and our ability to have anti-inflammatory and immune responses that operate properly. But there's always sort of been this gap in the literature where sometimes studies show positive results and sometimes they show negative results or null results or no results at all. And so really, I think here the microbiome is this intermediate that's meant to connect these gears here in the picture on the right. And so the gut microbiome was first defined by Joshua Lederberg, a Nobel Prize winning biologist. And he defined the microbes that colonize our bodies, both inside and out, as the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. And I think the key takeaways from his definition are that these microbes are commensal, meaning that they share space, symbiotic, that we both help each other, and that pathogens are there too. And we all right now, even if we feel as great as possible and know that we're in the best health of our lives, that pathogens are present. And that's important to keep in mind, because when you think about a community, think about the neighborhood in which you live. You may have some neighbors that you really like and some neighbors that you don't really like. And if the neighbors that you don't like are small in number, usually you kind of all come together and, grumble a little about them, but overall they don't really have much of an impact on anything. And so, but also that grumbling brings the other more positive members of that community together and gives you something to bond over. So I think having a little bit of everything in a community is important for kind of priming it, keeping it going, and kind of adding some novel things that, keep things interesting on a daily basis. So when we think about microbes, one of the big questions that emerged initially was, well, how many microbes are there in and on our bodies? And so initially, the literature estimated that we were outnumbered, actually, that for every one human cell that we had in our bodies, there were 10 microbial cells, because microbe cells are much smaller than the human cells. But actually, a paper in 2016 came out that disproved that, and it's not quite as impressive, but it is about a 1-to-1 relationship. So we're essentially half human, half microbe. And that's important when you think about our ability to metabolize things. So one of the most robust microbial communities associated with the human body is the one that lives in the intestine. It's the most diverse. It has more than 1,000 unique species, and it expresses a ton of different genes that carry out functions that human cells don't have. And so just to put that in perspective, humans can express 23,000 genes, but our collective microbial community in the gut can express on average 3.3 million genes. So they can do a lot of fabulous things that can either help our health or hurt our health. And when we think about where those microbes live, if we look at the bottom left, you can see I have some parts of the intestine circled here. So the small intestine is in orange, and there are 3 parts to that. There's the duodenum, the jejunum, and the ileum. The ileum is the last part of the small intestine, and that's really where microbes start to colonize. And then our food and digesta pass to the large intestine, and that's the green part. And then finally into the last part of the large intestine known as the colon. And that's really where we have a huge population of microbes doing all sorts of things to utilize additional food components that are there. And oftentimes they are producing things called short-chain fatty acids, which are really important fuel sources for our intestinal cells. They also have anti-inflammatory properties. They can travel around in the circulation and go to different tissues, and we'll talk a little bit more about that in terms of exercise and how those metabolites play a role between muscle and the gut. But if we look on the right here, we can see that I've outlined 2 things. Well, I guess a few mixed all together, but we have the microbes that appear on our tongue and the microbes that appear in our stool on the bottom. And if you look at the gray panels where it says microbes on the left, you can see that the tongue has a variety of different colors in comparison to the stool. And so this is just showing that throughout the body, the communities change, even though both of these sites are affiliated with the digestive tract. But if we look at the right column of that picture, you can see that the functions are actually fairly redundant. We have all the same colors sort of appearing in the mouth and in the GI tract. And that's important to note because oftentimes the literature so far is really focused on who is there and not what they're doing. And just because someone doesn't have a specific microbe in their gut doesn't mean that the function it carries out that's important for health isn't being carried out by a different microbe. So we could both be healthy because function Y is being carried out because you have microbe A and I have microbe B, but they're both doing the same thing. And so that's really a major gap in the literature right now, is trying to identify which microbes are doing what and how that translates to health or disease states later on. So because most of the literature has covered who is there, I do want to talk a little bit about that just so that you have a sense of how we organize ourselves and thinking about the microbes and identifying them. So if you think back to high school biology, you probably learned about the tree of life. And so that goes everywhere from domain to kingdom, phylum, class, order, family, genus, and species. And a lot of the work in this space has really focused on the phyla level, which is pretty high in terms of resolution. If we wanted the best resolution, we would be looking at genus and species level. And really, we're just, getting to that point in this scientific field. And so if you ever read about gut microbiome in the scientific literature, you're probably going to see a lot of the terms that are on the right here. So verrucomicrobia, proteobacteria, actinobacteria, firmicutes, bacteroidetes. And in this pie chart, the first thing you probably notice is that bacteroidetes and firmicutes are really large pieces of the pie. And in the Western world, where we tend to eat a very Western style diet that's a little bit higher in protein and fat and refined grains, a lot of processed foods, the ratio of bacteroidetes to firmicutes becomes important because that is the majority of our community. So we really want to be able to see at a lower resolution in those two phyla who's there and what they're doing. And if we just think broadly about what microbes would be doing if they're sharing body space with us, there has to be an evolutionary purpose for that. And so some of those functions that they carry out can be categorized into three main groups. So they protect us. So if you think about the intestinal lining as a monopoly board, the whole goal of monopoly is to buy up property and then kind of manage your property so that you make income. And so obviously there's no financial transaction here in the gut, but in terms of taking up real estate there, every little microbial cell that lines the intestinal surface is creating a barrier. And so they're protecting. When they take up a residential spot in the intestine, they're displacing another microbe from having that space. And so the hope is that all of our real estate is filled up with mostly beneficial microbes, and then pathogens get displaced, and then they just pass through and are excreted in our stool. This is also important because microbes will compete for different nutrients, and that allows them to maintain their space better because they're properly fueled. And in order to maintain their space, once they're established, they'll actually produce antimicrobial factors that also inhibit pathogens. Other functions include strengthening the barrier function. So this really aligns closely with protection. So when a microbe is there, it will release little molecules and chemicals that tell our intestinal cells to make stronger barriers between the cells. They can actually lead to immune cell production so that immune cells are ready there to fight any potential invaders. And we have tight junctions that connect all of our cells. And so those microbes release metabolites that foster the development of those tight junctions that glue all of our cells together. And then lastly, they nourish us. So when you eat dietary fiber, we don't have enzymes that break down dietary fiber, but microbes do. And then they produce these really beneficial molecules like short-chain fatty acids. And they actually make vitamins for us. A lot of our B vitamins and vitamin K are produced by gut microbes. And so that becomes important for energy utilization and other metabolic processes that keep us healthy. So going back to thinking about the gut microbiome then and how that relates to our current level of obesity-related diseases, we can see that over time, our modern disease burden has moved from infectious disease to non-communicable diseases like inflammatory bowel disease, allergies, celiac disease, etc. And so an earlier paper in this field sort of compared a healthy control and a patient with inflammatory bowel disease, type 2 diabetes, and then necrotizing enterocolitis, which is very specific to infants. And so you can see that within different health conditions, a healthy control looks different than someone who has that disease or condition. And so obviously this is just a snapshot, and there's a lot of inter-individual variability in what those communities look like. But just to give you a sense that if you have a disease or an illness, that can drastically change your microbiome. And it is a little bit of a chicken and egg question of which comes first. Did the microbes change and then you got the disease, or did you get the disease and then the microbes changed? It's probably a combination, a little bit of both. But that's important for us to think about. How do we treat and help people improve their health in the context that microbes are responding to the same cues that we are? So what do microbes respond to? What shapes them? And, in theory, when you are in utero developing before you're born, it's thought to be relatively sterile. And so your first inoculation of microbes really happens with birth mode. So were you born by cesarean delivery or vaginal delivery? Were you breastfed or formula fed? And then from there, what does your diet look like once you're grown up and eating solid foods? How do you exercise or stay physically active? We know that Some emerging literature is even showing that if you're just more sedentary versus, standing up at work and walking around, the microbiome looks different in those people. Do you have a disease? What age are you? Are you taking medications, especially antibiotics, which are typically not selective? So you may have an infection because of a single microbe, but antibiotics don't target a single microbe. They basically wipe out your whole community. And so that's important because you have to build that back up. And so after taking a medication or an antibiotic, you want to think about, okay, well, how do I get my microbiome back to where it was? And then also where we live. Do you live in a rural environment where you're close to a farm? Do you live in an urban environment? Etc. So all these things play an integrative role, and there's a lot of interaction between all of these things. So with that, let's take a closer look at how diet impacts our microbiome. So one of the first studies that really came out in this space and got a lot of media attention compared two communities. So both are children, but in Burkina Faso, this is a group of individuals who is very post-Neolithic, meaning that they eat a lot of grains and they just sort of started agriculture. And most of what they grow are grains like wheat, quinoa, farro, and things like that. And then they compared those children to an Italian group of children that eat a mostly Western diet. So higher fat, higher protein, more processed foods. And so if we just compare at the phyla level, looking at the microbes in these pie charts, you can see there's a lot of Bacteroidetes in these children eating more grain-based foods. And in those eating the Western diet, they have more firmicutes. So this paper really spawned this idea that Bacteroidetes are good and firmicutes are bad. And just for point of reference, that's a really broad stroke sort of summary here. And so again, remember on the earlier slide that I was saying we really need to look at a lower resolution and think about the genus and the species that are there and what they're doing. Because there's a lot of crossover. There are good firmicutes, there are bad Bacteroidetes. And so just looking at the phyla level like this is not adequate. But it is saying that as a broad class of microbes, they have a preference for certain types of food, but we need to understand what they're doing at a lower level. So when they did further analysis of these two communities of children, they thought, okay, well, let's look at these short-chain fatty acid production. And short-chain fatty acids are often fermentation products of dietary fiber. So as you would expect, you see in yellow here, more short-chain fatty acids in the children from Burkina Faso compared to those from Italy. And because short-chain fatty acids are acidic, it's sort of included in their name, acid is actually really helpful for deterring pathogens. So even though pathogens, you would think they're kind of robust and can cause infection in a variety of environments, they don't thrive in an acidic environment. And so that's one reason our intestine likes to harbor these short-chain fatty acid producing bacteria, because if you look on the right, you can see we have 4 different pathogens that cause different sorts of gastrointestinal issues. Salmonella should sound familiar, E. coli. And we see that in the Italian kids, there are more of these pathogens in green compared to those on the grain-based diet in yellow. So likely these short-chain fatty acids are deterring pathogen proliferation, which is a good thing. If we think about diet in terms of patterns though, so most people don't eat a single food in their daily diet. So we have, omnivores who eat a balance of animal-based foods and plant-based foods. We have this kind of growing popularity of eating more plant-based foods. And so I know that this looks like a terrifying diagram, but I've circled some parts here that I want to just point out. So on the upper part, you see the omnivores in purple, And then there's this cluster of lots of red squares. And so the red is depicting microbe groups that are positively associated with that type of diet. And so within that, we can see that Ruminococcus and Streptococcus at the genus level are being kind of upregulated. And conversely, on the lower half of the heat map, we see that the plant-based diet has less of them because the blue represents a negative association. And so then if we kind of scroll more over to the right on this picture, we see another little circle where I've highlighted some of the microbes that are increased in the plant-based diet, and those are Prevotella, Roseburia, and Lachnospira. And those three genera are kind of thought to be fiber fermenters. They utilize polyphenols from plant-based foods, and they're generally associated with health, whereas Ruminococcus and Streptococcus are generally more associated with negative health outcomes. So thinking about that, we can work our way around my plate and think about each food group and what's in it that our microbes might like. So with regard to fruits, we have a lot of different compounds, and I've highlighted 3 here. So we know a lot about polyphenols. There's coffee fiber, ellagic fiber, which come from berries and nuts and seeds. And in the vegetable group, we have inulin and fructans, which are a type of fiber. We have soy isoflavones, glucosinolates, which come from cruciferous vegetables like asparagus, broccoli, and cauliflower. More polyphenols, lignans, and then a lot of components that we can make short-chain fatty acids or SCFAs from. Within the grains group, we have a really diverse set of polysaccharides, so different sorts of starches that are slow degraded, where they may make it all the way to the colon and then get fermented by the gut microbes. We have resistant starches. We also have lignans and other oligosaccharides like fructans that can be fermented. In the dairy group, we do have some specialized fibers that can be made from milk sugars called galacto-oligosaccharides, and those actually appear naturally in human breast milk, which are really important for feeding a baby's gut microbiome. We have different fermentation products, so if you eat yogurts or kefir, which is a more smoothie-like yogurt drink, those are going to have fermentation products like short-chain fatty acids and other degraded carbohydrate materials. And many probiotics or fermented foods contain live bacteria, which are considered beneficial for health, like lactobacillus or bifidobacteria. And then lastly, we have the protein group, which this group is a little bit more complex in terms of whether or not positive metabolites or negative metabolites are produced. So obviously, microbes can help degrade protein down to amino acids, which are important for muscle growth and maintenance. There are components in protein that we can take, like phosphatidylcholine, which become choline, which is a type of B vitamin, which is important for energy metabolism. But depending on how you cook your meat, if you're like me and you really like charred meat off of barbecue, that's where you can introduce some other things that aren't so great, like heterocyclic amines and nitrosamines that happen from the charring of protein components. And obviously, everything in moderation is going to be fine in a diet. But it is important to just think about how often you're eating those things and the quantity, because also you get a lot of nitrogen in protein. And so nitrogen puts a lot of stress on our kidneys, and the microbes, have to help to process some of those nitrogen-containing compounds. So that's a lot of specific names and things that I don't expect anyone to kind of remember, but you can break this down into 3 groups that I call the 3 Ps. So when you're eating food, think about whether or not you're incorporating one of the 3 Ps or more that microbes really like. And so first, they like to have friends. So those are those probiotics. So if you can eat other live microbes like bifidobacteria and lactobacillus, then you're going to be adding more of a popular and positive member of the community into the gut. There are prebiotics, which are food for the probiotics. So these are going to be those resistant carbohydrates, galacto-oligosaccharides, and other dietary fibers like inulin. And then we have polyphenols. So these are those special plant-based nutraceuticals that have anti-inflammatory properties. So phenolic acids, flavonoids, lignans, and phytosterols. So thinking about protein first, and we'll work through each of the food groups here. This has been debated, I think, in the literature a lot in the last decade is, are high protein diets good? And there's really mixed evidence, and a lot of those studies, haven't really explored the microbiome. So my goal today is to take the very limited literature that is available and just share, in the context of microbes, is a high protein diet good? So in this particular study, they recruited obese males and they were trying to lose weight. So they put them on this phased approach where they were first on a weight maintenance diet with 85 grams of protein. After that acclimatization period, they switched to a high protein, moderate carbohydrate diet for 28 days. And that had 139 grams of protein. And then after that, they switched them to a 28-day diet of high protein, low carb. And so the protein stayed about the same, and then basically the carbohydrate just was shifted to the fat. And so in this case, on those higher protein diets, they did see increased concentrations of carcinogenic compounds like in nitrosos and a decrease in antioxidants. And they also saw a decreased production of butyrate, which is one of the most potent short-chain fatty acids that we know has beneficial health effects. And I mentioned Roseburia earlier as a beneficial microbe. That also decreased. And so obviously this is only one study, and we need to really look at different iterations of this to understand. Sample sizes are also small, and every person has a very individualized microbiome, and so we need to explore that more as well to capture that variability among people. But I think one thing that kind of emerged from that in another study that happened after that is, okay, well, maybe high protein isn't great for the microbiome, but what about if you switch your protein source? Because I mentioned, charred meat obviously is where some of those nitroso compounds come from. So what if you switch to a plant-based protein more so than animal-based protein? And so this study compared 3 different higher protein diets where the protein was coming from more red meat, more poultry, or more animal products. And it kind of showed that the overall diversity of the microbiome really wasn't dependent on protein source. It didn't matter. What seemed to matter was the content of saturated fat more than the protein source. And there were 19 microbes that they did find that seemed to be sensitive to protein source, but most of them haven't been well studied, so it's really hard to make any sort of hard conclusions about what that means. But I think what I take away from this is that, okay, well, diversifying your protein sources, getting a variety from both plant and animal-based foods is probably the way to go, and try to really get lower fat meat options if you're eating animal products. Switching to vegetables, I like to think about things in terms of how you can get the biggest bang for your buck. And when you think about vegetables, we kind of have 2 broad classes. We have allipaceous vegetables, which would be things like carrots, peas, celery. And then we have more cruciferous vegetables like cabbage, broccoli, radishes, cauliflower. And so I think that cruciferous veggies are probably going to give you more bang for your buck. Their colors are so deep and robust. And, the more colorful something is, the more polyphenols it's going to have. And so I think that really shows. This particular group of investigators published a study in 2009 looking at cruciferous versus allopaceous vegetables, and they found that the cruciferous vegetables really had an impact on which microbes were present. So you can see that there were some fiber degraders that were upregulated, Eubacterium hali, P. faecium, and A. putridinus. And interestingly enough, those microbes also correlate with polyphenol degradation. So cruciferous vegetables contain polyphenols called glucosinolates, which have anti-cancer properties when the microbes convert them to isothiocyanates. And so more cruciferous vegetables may have a number of benefits in terms of anti-inflammatory and immune responses. Other really deeply colored foods within the fruit category would be berries. So some individuals, tend to really focus on specific berries. And so cranberry extract is known to have some really nice benefits for obesity-related outcomes. And so in this animal model study, they put mice on a high-fat, high-sucrose diet. And when they added cranberry extract, they saw that weight gain was decreased, they had less adiposity, their triglyceride and inflammation levels also went down. And that seemed to correlate with improved insulin sensitivity and one specific microbe, which is called Akkermansia muciniphila. And it's one of my favorite microbes. And on the right here, you can see my little cartoon that I've made of him. So Akkermansia is a mucus degrader, and that kind of sounds gross, but when you think about gut health, as a metaphor or analogy for your lawn, so you mow your lawn and maintain it and, fertilize it, keep mowing regularly. Akkermansia is like a little lawnmower. He just goes through, and as the upper layers of the mucus in the intestine kind of get worn out, he trims them down, and that primes the cells to make more fresh mucus. And that mucus layer is really important for creating a barrier against pathogens. And it also is important for attracting vitamins and other nutrients to be absorbed. And so he is really important. And so berries might be a great way of fostering the growth of that beneficial microbe so that you are creating a nice, beautiful lawn inside of your intestine. And then thinking a little bit about grains in terms of prebiotics. So prebiotics are a special type of dietary fiber that are known to enhance the proliferation and survival of beneficial microbes in the gut. And so not all dietary fiber gets to be classified as a prebiotic. But there are two prebiotics that have been really well studied, and those are inulin and oligofructose. And most Americans consume anywhere from 3 to 5 grams of those on average a day. And believe it or not, the place that it's coming from in our diet is wheat products. And wheat is a really good source of both of these specific prebiotics. So these types of dietary fibers get fermented by microbes and turned into short-chain fatty acids like butyrate. And if you look at these pie charts here, you can see that other food sources in the American diet that would be providing these are things like onion, banana, and garlic. And obviously, no one's going to go around mowing down on onions and garlic because no one would want to be around them. So bananas and wheat are probably the two foods that we would consume more of. So, thinking about where these beneficial prebiotics come from, and what you can go to the store and buy that's going to be shelf stable for longer. Bananas kind of go bad quickly, especially in Arizona where I'm at. It's the heat just like sucks the life right out of your bananas. So this becomes important for thinking about where you can get the biggest bang for your buck. within the grain category, we can also think about whole grains versus grain components. So I remember when I was in graduate school, grain bran layers were all the rage. Everyone was like, oh, you have to eat wheat bran and wheat germ. But I mean, really, if you think about whole grains, you're getting the bran, the germ, and the endosperm. And so you're getting all those nutrients that naturally occur in that setting. And so if we were to compare bran versus just whole wheat products, the whole version has actually been shown to improve microbiome, beneficial microbes like bifidobacteria and lactobacillus better than an individual grain component like bran. Other studies have compared other grains, so whole grain corn versus non-whole grain corn cereals, and you can see that in both cases the whole grain and the non-whole grain increased bifidobacteria, but it was only significant in the whole grain cereal. But that's not to say that you should, not eat refined grain products if you enjoy them, but I would just try to have a balance of both. You need about 2 to 3 servings of whole grain a day is what we sort of recommend. And there's some literature to suggest that anything over 3 whole grain servings isn't going to add any extra cardiometabolic benefits for you. So that's nice if you're like, okay, well, I'm willing to eat whole grain cereal, but I really like my white bread. Well, have the white bread and just eat a few servings of whole grain cereal each day. And then thinking about which gut microbes then are produced. So again, wheat is a major staple in the US in terms of grains. And so there's been more research in that space. And so that's why I'm sharing literature about wheat here. So this study was really nice in that it tried to look more at the polyphenol content of wheat. So as you increase your wheat intake, you're getting more polyphenols, and there's a specific polyphenol in wheat called ferulic acid that gets metabolized and produces short-chain fatty acids, which have anti-inflammatory effects. And they are actually able to show that as you consumed more ferulic acid, you got this proliferation of Bacteroides and Lactobacillus, beneficial microbes, and that correlated with the decrease in inflammation. So these grain polyphenols are important for lowering inflammation, and they're also promoting beneficial microbes. And in this area, a lot of people are now eating gluten-free. And so I just wanted to take a moment to compare what a healthy microbiome looks like versus someone who has celiac disease, because that's an autoimmune disorder. And so in this case, in a healthy person, we have lots of different colors of microbes in our little monopoly board intestine. So that's showing us greater diversity. In a celiac patient, we would have less diversity. And you'll notice that all the real estate spots are taken up in the healthy patient versus the person with celiac disease. And so whenever you have open real estate, that makes an opportunistic chance for a pathogen to take up residence in that space. And so celiac individuals also tend to have more pathogens than a healthy person. And so this begs the question of, okay, well, that's a very different sort of intestinal environment, so I can understand why someone with celiac disease might need a different diet. But what happens when a healthy person eats gluten-free? And De Palma and colleagues actually did a study to look at this. They had healthy individuals eat a gluten-free diet for 1 month just to see what happened to their microbiome, and they found that a number of beneficial bacteria were decreased while pathogens increased, and that correlated also with a decrease in immune function. And so this is interesting because so many people, are sort of self-selecting to eat a celiac a diet that's really meant for celiac individuals. And I think there's even more information coming out now that most of these Gluten-free foods are rice-based, and rice is a grain that naturally likes to soak up more heavy metals from the ground. And so they've actually shown that people eating these gluten-free diets have more heavy metals that they're peeing out in their urine. And so we don't know if they're staying in their body, if they're all being filtered out, but not only is it changing the microbiome, but it also seems to be exposing them to higher levels of potentially harmful heavy metals like cadmium and mercury. Okay, so let's take a look at exercise. And this is sort of this fun area that's really just starting to emerge in the field of microbiome. So the majority of evidence is in animals at this point, but we definitely are seeing in animal models that exercise and sedentary mice have different types of microbes in their gut. We know that if you have animals on a high-fat diet versus a healthy control, and then you introduce exercise, that the exercise can sort of counteract some of the effects of that high-fat diet. And this actually corresponds with the picture here that you see in the bottom right. So ND means normal diet without exercise or with exercise with the EX. And then we have HFD, which is high-fat with and without exercise. And so right away, you see that the high-fat diet groups, whether they have exercise or not, kind of cluster on one side of the plot, and then those normal diets plot on the other. But what's interesting is that although exercise definitely is changing the community structure of the microbes, it isn't necessarily rescuing it back to just what a normal diet would look like. And so that tells us that exercise and diet have differential effects. And so they're each contributing their own unique positive contribution, and that having them together is maybe even having a greater impact because those are those green dots that have really pulled away from all of the other groups. And so, you know, in my field, I'm a nutrition person and I work with a lot of exercise people, and we're always fighting over whether nutrition or exercise is better. And these data for the microbiome tell me that you really need both to have the biggest impact. And then exercise also depends on the metabolic state. So if someone already has diabetes, you can increase beneficial microbes, but it seems to be a little bit harder than in a animal model that doesn't have a disease. And so we need to really explore that in humans. And one of the first studies that came out in humans was comparing elite athletes who played rugby to sedentary control humans that were either normal weight or overweight. And here you can just get a sense that the BMI is different, and then lean and fat mass are different, which you'd expect between athletes and sedentary, normal weight and overweight obese controls. And so if we look at the microbial community here and plot each person's community as a single dot in sort of multi-dimensional space, we can see that the rugby players are all the black dots, and the green and red dots are controls who have high and low BMI. And so regardless of BMI, if you're not an athlete, you kind of cluster together. And there's something unique about these elite athletes. They all kind of cluster over to the right. And so this group tried to figure out why that was. They did find that there was greater phylogenetic diversity, so how diverse in terms of evolutionary lineages you get with the microbes. And so the athletes were different from both control groups, but no difference between the controls. And what they really found was that protein intake was much higher in the athletes, which, you know, for rugby players, I could see a high protein diet being kind of more common amongst that population, whereas runners probably have a more high carbohydrate diet. But it really seemed that protein was driving these differences in phylogenetic diversity compared to the controls. And they further confirmed that, you know, creatine kinase, which is also a measure of protein degradation and muscle turnover, is also kind of driving that. In work from my lab, we've sort of just tried to look at just generally how active are you, and are there any differences in community structure? And so you can see these little lines with stars here in the MVPA plot. And that's just indicating that how close together groups with varying levels of physical activity cluster together in multidimensional space. So they're more similar in terms of community structure, and that as you increase activity, those clusters are each continuing to be unique. And then on the far right plot, we actually tried to see, okay, well, we know that diet and exercise have specific effects that are unique. And if we add in the dietary component like fiber, which we know is really important for microbial fermentation, we see that there are some differences there as well. And that one group is different from all the others, and that group is the high physical activity, low fiber group. So they kind of cluster differently to those that have different combinations of physical activity and fiber. And so we definitely, this is sort of just exploratory, and we're trying to figure out why that is, but this is all self-report and not a intervention. Other things with exercise are interesting in that you can cause a shift. So here at baseline, we have lean and obese animals, and then they put them on an exercise intervention for 6 weeks. And initially at baseline, they cluster separately, but then they start to, after exercise, look more similar. And then after a 6-week washout of no more exercise, they've actually maintained those differences. And so one of the really interesting things in this field is, if you intervene with exercise or diet, how long is it going to last? And so we don't really know the answer to that question. And obviously animals are very different than humans in some ways. And some of these changes were driven by certain microbes changing. But this is just sort of an introduction to sort of think about what that means and how long you'd have to exercise in order to maintain those effects. And I want to sort of end on what I think is sort of one of the most fascinating things in this field. And this is just to kind of prime you to think about how exercise and microbes are really related. So microbes make metabolites that can travel to the muscle, and our mitochondria are the components of muscle cells that make energy for us in the form of ATP. And butyrate, one of the short chain fatty acids, can go to the skeletal muscle and actually activate ATP production, and it can also cause brown adipose tissue to be much more active in burning energy. And so there's this, actually seems to be a crosstalk where microbes send these metabolites to muscle, and then muscle is sending cues back to the microbes. And the study that really found this link is they took fecal samples from elite marathon runners, and then they fed the microbes from the feces to animals, and then they put the animals on a treadmill, and they saw how long it took them to run to exertion. So athletes have these endurance athletes had greater abundance of specific types of bacteria. And in particular, they had a species called Veillonella atypica. And that microbe actually is associated with this greater performance in athletes. And it was also associated with greater performance or longer performance in the animals that ate these microbes from runners. And what they actually found was that when muscle produces lactate, that lactate can actually travel to the gut through the intestinal cells to the microbes, and then they will take that lactate and turn it into short chain fatty acids, which then travels back into the blood to the muscle, and then further allows for this metabolic cycle. So whereas we think of lactate as sort of building up and causing fatigue, the microbes are actually helping to manage that metabolite so that you can run longer. So I don't know, in the future we may see Veillonella atypica probiotics or something for athletes, which will be really cool. I know this group is working to commercialize a probiotic for that. So what should you be telling your clients? There are so many amazing things in this field, but it's still very, very young. So I think just talking to them about exercise and diet, that both of those can result in different sorts of community shifts, and that both, contribute something important. And that change is hard. So making 1 or 2 small changes at a time is probably the way to go. So if they're more willing to start with exercise or more willing to start with diet, work within the constraints that that person has so that they can be more likely to be successful. And one thing I tell everyone is that, I don't know, in the US at least, we're very weird about bodily functions in public. And so rumbly tumblies aren't always a bad thing. That, a little gas just means that your microbes are saying thank you for feeding them something that they can ferment. And so, trying to shift the paradigm about how we think about GI symptoms, and obviously within reason, like nobody wants explosive diarrhea or anything like that. And that really how exclusionary diets might be contributing to that. So you may pick a gluten-free diet because it makes your stomach feel better, but what if it was actually that your stomach was just, or your intestine was allowing for some fermentation of those fibers and your microbes were really liking it and they were telling you they were happy, and now you've taken that away from them. So I think encouraging moderation rather than full elimination of food groups is probably the way to go. And with that, I'd like to thank you so much for joining me today, and I'd be happy to answer any questions that you have.
All right. Well, thank you, Corey. This was just fabulous as always. I don't know if anybody else out there is feeling like, whoa, there's a lot going on inside me. I had no idea that I had all that biochemistry happening. There is a lot going on. And I think one of the things that I'm always reminded of when I hear you give this presentation, and I've heard it a few times, is we often think that we're trying to be healthy by doing something, whether we are, reducing one dietary component, increasing another, taking a supplement, doing something else. And the impact can be much more than we imagine because we are only beginning to learn about how everything we do influences this colony that lives within us that seems to have such a dramatic impact on everything. So I don't know, as a dietitian, it's fascinating. So I can tell from all the questions that you guys have been asking that you're also fascinated. And so let me get right to these. We had a number of questions, Corey, about, let me just start with the first one. We'll go back to the very beginning. Tim asked, for those with celiac, are there certain other grains that encourage Bacteroides or typical wheat replacements that have a more negative impact? I think what he's getting at there is if you can't eat wheat, what are some good grain alternatives?
No, yeah, there's definitely a whole slew of things that you could opt for. So I would probably go for something that has high levels of beta glucan fiber. So oats would be a really great alternative, and most oat millers have sort of moved away from sharing the same milling path for wheat and oats. So you can be assured that most oats are going to be clean of any sort of gluten contamination. Other grains that are really going to give you some extra polyphenol bang for your buck would be things like quinoa. It also is high fiber, but doesn't have gluten, so that would be a good option. Bulgur, oh, sorry, that's wheat. Ugh, sorry. Brain malfunction. I would kind of think like buckwheat is probably going to be good. It's just when you buy processed foods that say they're gluten-free, they often are rice-based. So look at the packages and just make sure that you're balancing those with some other grains that you're introducing, and you'll be fine. So don't avoid them altogether, but just make sure that that's not all you eat are those processed options.
Okay. All right, here's another good one. Matthew asks, how would a dietary substitute such as almond milk compare to traditional dairy for microbes?
That's a really good question. So a lot of these milk alternatives are not super well regulated in terms of like what nutrients need to be in it. So the first thing that comes to mind, and this isn't necessarily microbe related, but cow's milk is fortified with a lot of vitamins, and it's really high in calcium naturally. The one thing that you have to look at for any of these nut milks or grain milks are that they're not required to fortify. And so oftentimes, if you don't look, they're not going to have calcium in high amounts. They're not going to have a lot of the vitamin A and the vitamin D that you would get from cow's milk. And so look for that. Find the brands that are actually trying to fortify to be equal to dairy. And really what you're looking for is that fermentation process. So you can get fermented soy yogurt as an alternative. Those are still going to give you the same benefits. Obviously, the microbes might be a little different for what's optimized for fermenting soy, but it's the actual fermentation and the live microbes having those there that's important.
Okay, all right. Kelly asks, how do oxalates play a role in the microbiome?
Yeah, that's a good question too. So oxalates will bind up things like minerals and block them from being absorbed in our intestines. And they do that binding mostly in a neutral pH environment. And so if you're eating fiber, which also tends to come with oxalates and phytates, you are giving the fuel to the microbes that they can ferment. And when they produce acid, it forces the oxalates and phytates to let go of the minerals so that you can actually absorb them. So it's sort of a double-edged sword, really. I mean, because fiber and phytates and oxalates come together, but at the same time, if you're getting fermentable fiber in the diet, you're actually going to allow for production of a beneficial environment so that they don't cause as much of a problem for you.
Okay, good answer. All right. I believe this is Michael asks no. Yeah, Michael says the study regarding high protein diets. I think that was early in the presentation. Were those animal proteins, plant proteins, or both?
So the first one that compared the 3 different phases where they ramped up protein, it was primarily animal. But it did have some plant-based. The second study I focused on really tried to cause major shifts in the protein source so that you could actually delineate what was doing what. So the first study was it was more animal-based.
Okay. All right. Kind of along these lines, Matthew is asking, in regards to the Impossible Burgers, is there any research on how it compares to a normal burger? And I think he's referring in terms of how it affects the microbiome.
I haven't seen anything, but that doesn't mean I didn't miss it in the literature on how the Impossible Burger affects the microbiome. Mm-hmm. But in terms of micronutrient composition and then macronutrient composition, they've really worked hard to make the Impossible Burger meat really comparable to actual beef. So it's got the same iron content, it has the same protein and fat content, plus or minus a few grams. But I think in terms of a sustainability-based product, they did a really nice job of trying to make it comparable. Now, because it is plant-based, my guess is it would probably have a lot more of those like prebiotics and polyphenols that are going to be more beneficial for the microbes than animal-based protein.
Okay, good answer. All right, so we've got time for one more question. I'm going to combine 2 people's questions here. So this is a question from Angela, and also I'm combining it with someone else's question, Michelle's question, and this is about for someone with celiac. Should they just take a probiotic, prebiotic to increase the diversity of their microbes? And kind of as a part B to that question, if they do take a supplement, are refrigerated probiotics or shelf-stable tablets better?
I definitely think any autoimmune condition could benefit probably from taking a probiotic. And if you're going to take a probiotic, I would recommend also having the prebiotic there. And I think in this field, like in the clinical setting, you're starting to see, if you go to the pharmacy at Walgreens and look at the probiotic section, many of them are actually what we call symbiotics, where the pro and the prebiotic are merged into the same pill. And so that's going to be important, because just like if you have a little kindergartner and you're putting them on the bus in the morning, you're not going to send them without their lunch, right? So if you're going to send a live microbe into the acidic environment of your stomach that's like a super, super low pH, you probably want to send some food so that they can sort of thrive and make it to the intestine. With that, each condition that you have, it seems that a different probiotic is going to be beneficial. And this is an area where we don't know the answer super well. And so I would probably just, try one and see if it works for you, because as far as evidence goes for celiac, I haven't seen anything where they're recommending a specific probiotic for that yet. But other inflammatory bowel conditions, there are some pretty strong links. Crohn's is sort of a mess in terms of they don't know which one works. That said, refrigerated versus shelf stable, I think what's more important is it a reputable brand that's been around a long time? So I don't have any affiliation with the company, but they've been around a long time. Culturelle is shelf stable. They've done tons of research. They know their products work, and they do a lot of testing to make sure that the probiotic is viable and shelf stable for the period of time that it's guaranteed. So I would probably opt for one of those, and they already come with the prebiotic added. And then you don't have to worry about having the proper storage, especially if you want to go on a vacation, a weekend getaway somewhere to a cabin. You can just take that shelf stable pill with you. It's super easy. So, and it's cost effective. I think the refrigerated ones tend to cost a lot more, and I'm not sure that you're getting anything extra out of those.
Great answers, Corey. Thank you so much. And thank all of you for joining us today. I think if nothing else, clearly this is a very emerging science, and it is also, there is a lot of information and disinformation out there. If you are seeking more sources, and Corey, I would ask this of you too. I mean, if someone wants to know more about the microbiome, what should they do? How can they learn more?
Well, there are tons of media articles out there. And so I would just encourage you always to, if you read a media article, see if it has the link to the actual scientific paper. Try to take a look at that, because sometimes the framing is a little awkward and doesn't really give the true essence of what they found. There are a number of sites where you can go for some really great information. So the International Scientific Association for Probiotics and Prebiotics is a really great resource, and they have great advice on different prebiotics and probiotics to take for different things. Mm-hmm. That's probably where I would check first, because they do a good job of maintaining their website. And then other places to look would be Healthline.gov. They do a nice job of updating and actually doing a nice way of citing current literature. So those two sites would be places I would look first.
Great. Thank you. All right. And I will just remind everybody that we have uploaded the CU certificate onto the files section of the webinar. And there are a number of other handouts. So I hope you've gotten a chance to download those. Again, thank you all for participating. We really appreciate it. Thanks for your great questions. If you do need to reach us, that information will be coming in the follow-up email that you'll be receiving shortly. Again, thank you for everyone. We'll look forward to interfacing with you in the future. We certainly will be bringing more great webinars like today's. Thanks so much.